All right, so today I was planning on actually talking, so I was planning on sort of covering sort of at a little bit more superficial level, the uh, uh, contrast processing, acquisition and applications. I had hardware there, but I don't know why I put that. It's actually more acquisition. And instead, I decided just as I was making my talk, I decided that I was dwelling more on, I think, the important aspects, looking at the rest of the lectures. I decided to actually focus mostly on bold contrast and on ways of acquiring it, of the dynamics, of the spatial patterns of bold, of everything about bold. I'm completely fascinated about the dynamics and the patterns and the information that can be contained in it. So I'm also talking at the very end a little bit about perfusion and a little bit about blood volume. So, but just to give you a perspective, just to start out at the very beginning. So the brain at baseline, there's a certain level of CBF. Blood volume is about 4%. The blood is about 4% in capillaries. The metabolic rate, there's an ongoing oxidative metabolic rate that continuously happens because of this to power the sodium-potassium pump. I'll get to that in a second. You have arterial oxygen, which is almost fully saturated with oxygen, and the venous oxygen is about between 60 and 40% saturated because of the metabolic rate that pulls the oxygen out of the blood. So looking at neurons, the resting membrane potential of an individual neuron, you probably learned this in basic neuroscience, is minus 70 volts. And there's basically high potassium on the inside, low sodium on the inside as well, and other negatively charged protons and organic ions and ions. So outside there's high sodium, low potassium, and chloride. So you have the sodium-potassium pump keeping this gradient to some degree. And during an action potential, and this lecture is not about action potentials, but essentially, once the membrane potential receives excitatory input such that it goes to about minus 55 millivolts, then a cascade of things happen. I mean, this is pretty much the basis of everything going on in our brains right now. And it's essentially, and it's a really efficient way of transferring information. So you have the neuron action potential, and essentially the sodium channels open, sodium rushes into the cell down an electrochemical gradient, and the membrane potential increases to 60 millivolts. And then you have voltage gated sodium channels that inactivated, and then voltage gated potassium channels that open. Potassium flows down the concentration gradient, then carrying positive charge outward, making the inside more negative again. So this is all a very brief cascade of events that takes on the order of about 10 milliseconds or less. And it basically recovers pretty quickly as well. So that is sort of like, and there's a certain refractory period, and basically that travels along dendrites or other nerves that actually have either myelin or whatever to transmit information all over the brain. And the likelihood of this firing or not, depending on whether it's inhibitory or excitatory, all determines how information is exchanged. So what accompanies brain activation? So there's spiking of local field potentials. So the spiking, as I mentioned, the spike rate is about 5 to 50 spikes a second during rest. And I'm sorry, that actually increases 5 to 50 spikes per second in the area that's active. It's typically transient, so there's a burst of spikes and then it goes down to some level of constant spiking. It falls near baseline. But the population average is it goes up, in the whole population of spikes, about 2 to 10 times. The local field potential, which reflect synaptic and membrane currents and network synchrony, They consist of several different types of information. There's low frequency, less than 10 hertz, which is delta and theta. That has slow cortical and hippocampal rhythms. That includes slow cortical and hippocampal rhythms. It basically phase modulates the spiking, and the power reflects the brain state, either sleep or navigation. So then you have alpha frequencies, 8 to 12 hertz. During activation, that typically goes down. It's an inhibitory rhythm, so it goes down, meaning the areas are excited, are allowed to be excited more. It modulates excitability and information flow. So beta frequencies, 13 to 30 hertz, also go down typically. And they basically inhibit motor control, motor activity and motor control, or they are involved with motor control. So transient bursts reflect mesoscopic dynamics, so like intermediate length dynamics. Gamma frequencies, what we're probably most familiar with, are during wakefulness. If you're attending to something, if you're doing a complex task, you usually have gamma frequencies, 30 to 80 hertz. And they go up anywhere from 20 to 100%. And they actually track spiking pretty closely. So they're the one frequency that seems to track spiking. And then at the very high end, you have, if you're sleeping, you have hippocampal ripples. That typically, no one, I mean, the bottom line, no one knows exactly what any of these are doing, but this is an estimate. They typically, with hippocampal ripples, they're replaying memories, and they're involved with consolidation. They speak to the rest of the cortex while you're sleeping, or even a little bit during wakefulness as well. Okay, and so bottom line, if you want to remember something from this, is that the local field potentials capture population level oscillations in synaptic and subthreshold activity, while spiking kind of sits on top of that and is modulated by these rhythms. All right, this chart is kind of intimidating, and actually this is what I spent most of the time working on for this talk. And it's basically why is it telling me? Oh, it doesn't show up there. So this is actually all that accompanies brain activation that one can measure with MR. So I'm just going to take a little bit of time and go through this chart. So to begin with, you have, and it's actually ranked according to how sensitive you are to the physiologic measure using MRI, at least. So I'll get into deoxyhemoglobin. So the reason why bold contrast is so successful in humans is that it's incredibly sensitive, or at least the signal is extremely robust. So relative to all the others, if you're ranking them from 100 down to 1, this is 100. This is the most sensitive. Contrast to noise in a typical time series is about at least, so the noise level is about 1 to 2%. The signal change is about 5%. So that's either 2 to 1 to 10 to 1, something like that. Can get up to 10 to 1. And it's a function of field strength, as I'll get into. You also have measures of CBF, cerebral blood volume. During activation, the flow velocity actually in the middle cerebral artery goes up slightly. And that's very slight. And you can measure it with phase contrast MRI. You also have changes in CRMO2. And changes in CRMO2 occur, that they can actually be measured by looking at the ratio of flow to bulb. And to get a measure of how much oxygen is extracted from the blood. So then you have venous oxygen saturation. So one would think, oh, isn't that bold as well? So it's actually not. It turns out that bold is mostly sensitive to the amount of deoxyhemoglobin. It's not sensitive to the amount of oxyhemoglobin or the amount of oxygen in the blood. It's just the amount of deoxyhemoglobin. So I put this here though, because one thing nice about preparing for these talks is that I look in the literature and I find that there's a lot of techniques, some that were presented at OHBM this year called zero TE equiplanar imaging, which is really, really nice techniques. And they're always puzzling over the contrast. And they thought, well, maybe it's inflow effects, But they also suggested that it could be T1 changes, longitudinal relaxation changes, due to dissolved oxygen in the blood, changing the blood T1. So it's not certain, but it's an interesting idea. So I put it down. Other things, and this is basically looking mostly, these next few are looking at spectroscopy. Spectroscopy is actually very sensitive to a lot of different metabolites. And I'm not going to go into all of these in detail. There's also GABA, basically an inhibitory neurotransmitter. You have glutamate, an excitatory neurotransmitter, if you really want to get into that study. Although you should note that, the sensitivity relative to the bold is about 1/20th that of bold. That's pretty much why it's not used that much. It's really hard to get a robust signal in a reasonable amount of time. Are looking at cerebral spinal fluid flux. That's sort of a new hot topic, the idea of looking at the glymphatic system that flushes out the brain when you're sleeping. But another idea is that with brain activation, the CSF has to go somewhere. There's an increase in blood volume, and tissue is pretty incompressible in the brain, so it pushes out the CSF in the areas that are active. And there's some groups that have noticed this. It's something I think worth exploring. Other things that are worth exploring potentially are, we're looking at Tyler Morgan is looking at sodium flux. So basically sodium, as I mentioned at the very beginning, rushes into the cells. And when it rushes into the cells, the relaxation rate of sodium changes a little bit. And it changes enough such that one could detect it using sodium coils. You can't detect it with standard proton MRI. So that's another technique that's sort of exploratory. People have explored, I skipped one, people have explored looking at brain elasticity. The brain changes its elasticity during activation. And the reason is sort of, it's less interesting than you think. It's basically the vessels become filled, if they're filled with blood volume, they become more rigid. And so that area becomes more rigid as it's with brain activation. All right, so then you have other metabolites, transport shuttles and things like that, aspartate and NAD, creatine kinase. But then beyond that, you have temperature. So the brain actually gets a little bit hotter, or more, the temperature increases a little bit during activation. And some people have been able to measure this either using diffusion imaging, so the diffusion coefficient changes slightly with temperature, but also phase contrast imaging, very sensitive changes in the resonant frequency of water with temperature. So that's another technique. It's extremely insensitive, but it's something that's there. So then you also have cell swelling. So that's actually a technique that Denis Lebihan, who was a pioneer in diffusion imaging, claims that he was able to see. So if you have a lot of diffusion weighting, so a lot of you probably haven't learned about diffusion weighting yet, but the idea is that you're very sensitive to you're able to, the signal is proportional to the rate of diffusion of the water molecules as they randomly move in space. So with a high amount of diffusion weighting, you're very sensitive to the water molecules. The higher the amount of diffusion weighting, the slower the diffusion rate that you're sensitive to. So the idea here is that during activation, neurons swell, then there's more intracellular water, and around the membranes of the neurons on the inside, the diffusion coefficient is a little bit less. So you have a larger fraction of lower diffusion coefficients and spins. So then that basically causes a decrease in diffusion coefficient or an increase in the signal if you're diffusion weighting. So a lot of people have tried to look at this, including our own group, and we haven't really found it that robustly. And some people claim it could be more vascular. It's one of these things that people throw out there. some people see it, most people don't, and it kind of like lives in sort of an existence of, it's sort of there, it's never been disproven, it's never been proven. And finally, and there's also neural current imaging, and that this sort of appeals to physicists because it's sort of the same source of signal for MEG, magnetoencephalography, the dipoles, basically current along the dendrites. So when you have a current, you basically create a magnetic field around the current. And so that magnetic field then superimposes on top of the MR field and creates distortions. And just like around susceptibility gradients, around red blood cells and so on, these distortions might either dephase spins or cause a phase shift. So theoretically, it has to be doing that. Theoretically, it's doing that. It's very transient. It's during an action potential. However, if you do most of the back of the napkin calculations say that the amount of phase shift or magnitude change that occurs with this is at least an order of magnitude lower than what we can detect. And so I put that as, relative to bold, which is 100 sensitivity, this is 0.02. So it's pretty low. And I don't even know if it's that, because nobody really has detected it yet. So there might be a way of pulling it out better, but that's that we'll find out. Okay.
One was about the previous slide.
What's that?
So why do you put a star for the second one, CBF? It's 25.
Oh, yeah, I put a star. And the reason why I put it, thank you very much. Very observant. All right, so that's good. CBF is generally 1/4 the sensitivity of bold. But I put the star there, and I'll show a few towards the end of my talk, if there's anyone hanging around. It's because of the fact that CBF measures are so stable, temporally, so incredibly stable over time, that if you are doing a long study where you're actually having activation that lasts 5 hours, let's say, or maybe even an hour, so you steadily on for an hour, bold signal will become, you never design bold paradigms this way. What you usually do is at the most, you have a block design of like 20 or 30 seconds. And the reason why you design block designs that way is because if you have inevitably the baseline will drift. And of course you can do your best to correct it mathematically, or else try to minimize the drift in some other way, but it will drift. And if it does drift, if you have this oscillation against this background of the drift, it's very easy to pull out. It's very easy to separate mathematically. If you have a long activation and then with drift, there's no way of really separating those two in any sort of way that you're certain. If you don't know, so with ASL, it turns out, and I'll show a study where it's rock solid. So if you compare, because it's basically a constant difference between one image to the next, where you have with a tag where the blood flows in, without the tag where it's not flowing in, and you're constantly subtracting, and you're getting a quantitative measure of one parameter, and that's blood flow, blood perfusion. And so you don't have any of the background signal. And so that's why it's just rock solid. So you can actually do a perfusion study. If you've registered the brain perfectly, you could do a perfusion study one day and then do the control the next day. You can do activation one day, control the next day, do a subtraction, you'll get a nice perfusion map of the difference. If you tried that with bold, where you did, let's tap your fingers today and let's not, there's no way you'll get an activation map. So that's why that star is there. It's 1/4 of bold, except for in certain paradigms where you wanted to compare long-term effects. And there, it's probably comparable to bold. The contrast noise doesn't go up any, it's just that bold is so bad at certain long time series. All right, good. So that's, I wanted to give that all up front because that's sort of the big picture. And I'm mostly going to be talking about what's in the box there, just these few, and mostly bold contrast for the most part. Okay, so once again, just to keep on hammering the nail in, this is a nice schematic of during activation. So just to review what I talked about last few days ago, you have oxygenated blood, which is bound to all the iron is bound to oxygen. And when it's bound to oxygen, there's no free radicals. And it's the free radicals, the negative charges that actually focus the field and distort it. So if there's none there, it's diamagnetic, and it's essentially invisible, like the rest of tissue. It sort of blends in. If it's deoxygenated, it has free electrons that attracts the field. It's less diamagnetic. It's pairing the individual iron molecules become paramagnetic, but the red blood cell is less diamagnetic than the rest of tissue. So it's diamagnetic, but it's less diamagnetic. So it distorts the field. So during activation, during rest, you have some sort of signal that's down. During activation, you have less deoxyhemoglobin that's present. So flow, volume, and oxygenation go up. And I showed this chart before. This cascade of events: cerebral tissue activation, vasodilatation, an increase in cerebral blood flow and volume. The oxygen delivery exceeds metabolic need. I talked a little bit about that we still don't know exactly why it should do that, but it's mostly either to deliver the oxygen if it's diffusion limited or remove waste. Increase in capillary venous oxygenation. This decreases the amount of deoxyhemoglobin, and which is paramagnetic versus diamagnetic. And that decreases the amount of susceptibility related intervoxel dephasing, which then increases T2 and T2 star, and that causes a signal increase in pulse sequences that are T2 or T2 star weighted. T2 star is transverse magnetization, essentially. So I'll get into that a little bit later too. Without actually showing a pulse sequence diagram, I figured I'd show a schematic of a pulse sequence diagram. And essentially, when you collect your images, you use what's called echoplanar imaging. If you've been to the scanner, you hear the beep. That's actually the gradients oscillating extremely rapidly to spatially encode the spins. And is that an RF pulse, radio frequency pulse is given, typically 90 degrees to knock the magnetization into the transverse plane. And then what happens is that immediately, because the spins are experiencing different magnetic fields, this starts to dephase. The signal starts to dephase. Each spin is experiencing its different frequency, so it goes out of phase. And the signal, which is very much a function of how all the spins add up with each other, quickly decays. And the decay can be modeled quite nicely by an exponential. So, and during this decay, you want to collect all the data before the signal dies away completely. So you have echoplanar imaging. And this readout window is about, 20 to 30 milliseconds, typically. So the terms here is that the signal is an exponential decay. It starts out with the initial amount of magnetization. And you have the echo time, you have T2 star, and a lot of people use R2 star, which is just simply 1 over T2 star. It's the decay rate as opposed to the decay time. All right, and so this is an important concept I want to show you. This is actually taken right out of a few of these slides are taken out of my PhD thesis. So even though it's old, but it's nice for illustrating basic points. So this is basically a model of some signal. And as I just showed you, the signal, if right after the RF pulse, the signal, let's say in gray matter, dies away. And it dies away. This happens to be a T2 star 50 milliseconds during activation, and you can barely see the difference in the curve. It's 48 milliseconds during rest. So there's hardly any difference in this relaxation rate. However, if you take these two exponentials and subtract them, what you find is that if you find that they peak, the difference in the two exponentials peaks at about where the TE, the echo time, equals the T2 star. So this is actually the echo time. When you set your echo time on your pulse sequence on the console, it's typically about, well, this is actually a simulation meant to be at 1.5 Tesla. So at 3 Tesla, it's about maybe 30 milliseconds. So that's where that peak is. That's why you set it there, because that's where the maximal contrast occurs between the two exponentials. One very important point is that if you look at the percent change between the two exponentials, that will just keep on going up. So you'll keep on getting a higher and higher percent signal change with a longer echo time. And some people think, well, why don't I just keep on having a longer echo time? I'll get a higher percent change. Well, because of this curve. It's that what really matters is the contrast, which is the difference, not the percent change. So that's why you focus on having an echo time right here. Because with here, the percent change goes up, but then the noise floor also goes up and the signal just dies away. All right. All right, this is a little bit more involved, but this is basically looking at the field strength and echo time effects on contrast. So 3 Tesla is better than 1.5 Tesla. 7 Tesla is a lot better than 3 Tesla. And the reason why is because the change in T2 star goes up. So when the change in T2 star goes up, then you have more contrast. One problem, though, is that at higher field strengths, the baseline T2 star goes down a little bit. So if it went down a lot, then there'd be no advantage of going to 7 Tesla. I mean, certainly, yeah, you have higher, you have more signal overall, you have a little bit more sensitivity, but in terms of contrast to noise, it would be about the same. But luckily, there's an advantage because the T2 star doesn't go down quite that much. The baseline T2 star doesn't go down with high field. So if you just look at these curves right here, this is at 1.5, this is at 3, this is across essentially different baseline T2 stars. So if you're in an area, this is actually tries to illustrate this point. If you're in an area where the signal is dropped out, where there's a big hole in the brain because of a shimming problem, and if you try imaging there, you don't want to be at a TE of 30 milliseconds. You want to be at a TE of 10, because looking at this contrast versus echo time curve as a function of baseline echo time, the peak occurs wherever the baseline T2 star is. So that's hence the advantage of some of these zero echo time sequences that, around the brain, you have a huge difference in T2 star all over the brain. And on average, it's about 30 milliseconds. And that's why we use 30 milliseconds. But so that just sort of covers that part. All right, there's different approaches. And this is sort of getting a little bit more into the weeds, but it's just to give you an overview that there's, people play with different strategies for collecting the data. So when you collect your data, it's often depicted in terms of what's called K-space. And that's, you'll hear more about this later, but basically that's where each oscillation of the gradient that you hear in the equiplanar readout is essentially a path that goes through K-space. And you fill up this K-space. This is basically the inverse Fourier transform of the image. So you collect your data in this frequency space, and you create an image by making the inverse Fourier transform of that frequency space into image space. That it outpops an image. So the edges have high frequency, the center is low frequency, and there's different ways of going through it. So and it's how your image looks depends very much on how you go through the K-space, not only how it's filled up, but how you actually traverse it. So we use single shot equiplanar imaging because we're able to fill up this K-space, this raw data space, in a way that freezes all physiologic motion. And that's critical, not only because it's fast and you collect a time series of images, but because physiologic motion can alter the phase of any one line of K-space. And if these lines are sort of misaligned a little bit, then it will cause ghosting in your images. And that's okay, but the ghosting will be different every single image. So with equiplanar, you might have some imperfections in K-space, and that causes ghosting, but the ghosting is rock solid the same every image. And that's key. So, there's other strategies. There's spiral scanning, which you go from the center out, which is great. But there's artifacts associated with each type of approach that are different as well. But we typically like flipped echoplanar imaging, kind of zigzag through K-space here. All right. There's other methods for going to much higher. Yeah.
So you have the imaging for spiral. Are there times where you might prefer doing 2D, like a 2D image versus a 3D image?
Yeah, I didn't even get into 3D, but I generally, so yeah, that's sort of a involved question. I didn't even mention 3D. I was planning on skipping over that, but 3D is basically where instead of doing having a slice select pulse and then going through K-space one slice at a time, with 3D you basically do phase encoding and frequency encoding and slice encoding all at the same time, pretty much. And I think it's good, but I think it also has a few more phase errors involved with it because the amount of time for 3D to be collected, see, with one slice at a time, at least you have each slice is collected completely self-contained within 20 milliseconds. With 3D encoding, you can traverse different segments of K-space in 30 milliseconds, but line up is a function of where you are, like in the cardiac cycle, where you are breathing. And so they can have phase errors, and you could have non-reproducible, non-repeatable artifacts over time. And there's ways of correcting that. I don't know if that's the answer that you were expecting, but people generally, but here's the one thing. So, all right, I'll add one more nuance to that. That is, people typically do 3D imaging to go to really high resolution if they want to have a whole volume at very high resolution. And when you go to high resolution, your signal to noise is low enough such that physiologic motion induced phase errors and things like that are about on the same order as your thermal signal to noise. So it's less of a big deal that you have that sort of effect. So that's why people like to do 3D. No one does 3D imaging at like low resolution. They do 3D imaging at only high resolution. So, all right. Okay. Just very briefly, and this is probably way more than you want to know, but it's important to at least see. When you go to the scanner, and I don't know if some of you might be operating the scanner, you press, you can specify which way you want your read direction to be, left, right, AP, up, down, and depending on how you do that, you'll have different warping in the image. So there's one main concept that I want you to learn here, and that is, or two concepts. One, there's two major types of artifacts, aside from ghosting. Ghosting is another one I'm not going to talk about. Two major artifacts in echoplanar imaging. One is signal dropout, and the other one is warping. And both of them we haven't figured out how to solve completely yet because we can't get perfect shim on the scanner. So you usually press a shim button that tries to shim as well as possible. Because there's so much detail in how the fields are inhomogeneous, like for instance in the sinuses and so on, that having a perfect shim is very, very, very difficult. We get close, and at high field this becomes magnified. So if you have, so your signal dropout is very much a function of your voxel size and also field strength. So the larger the voxel, the more likely it is that you'll have more spins or more protons that will be processing at different frequencies, and they'll get all out of phase and the signal will drop out. They will cancel. However, if you make your voxel smaller and smaller, you have more of a homogeneous distribution of spins because you're sampling less. And so it's more likely to be in an area where the field is the same. So that's dropout. And also warping. So warping, so now getting back to the initial thing that I was talking about, where depending on the direction of your readout. So when you collect echoplanar data, your the way you fill K-space is very different, depending on whether you're going left to right or bottom to top. So if you, let's say, raster it, this is color coded right here, from bottom to top, it actually takes about 0.25 milliseconds to collect each line left to right. So that's really fast. And it takes about 30 milliseconds to go bottom to top. And so essentially, because it takes so much longer to go bottom to top, you'll have much more time for spins to get out of phase going in that direction. So the warping will be in that direction, essentially. So this is where KY was in the AP direction, and this is where KY was in the left-right direction. You can see the warping is totally different. Yes.
So here you're going slice by slice, right?
Yeah, it's just one slice. I'm just talking about one slice, right? Yeah. And it's different for 3D, but I'm not going to get into the details of the physics for 3D. So, and the reason why people always like to compare to structural images, let's say, instead of equiplanar images, they say, oh, well, we compared it to the structural image, and that somehow looks perfect. Why does that look perfect? Because if it's multi-shot, if you collect one line of K-space at a time, then essentially the time it takes to traverse that from bottom to top of K-space is essentially infinitely short, because there is no time that you allow to accrue between one line and the next and the next and the next. You're collecting one line at a time. So so that there's no distortion in one direction, and there's only a tiny little bit of distortion in the other direction. So 0.25 milliseconds time, essentially. So that's why your structural images look great and your equiplanar images look bad, because of the length of the readout window. And if you try to squeeze the readout window to be shorter, then the images will look better and better in that regard. All right. So, all right, I'm actually, I think that I'll just go till 2:15 and then finish the other part of my talk. I'll fit it in before the next one somewhere. I'll let you know. But I think I'll naturally just go to the end of bold here. So I don't want to keep people too long. All right. So now let's compare spin echo versus gradient echo. So there's different ways of looking at bold contrasts such that you can be sensitive to small compartments or large compartments. So with gradient echo, equiplanar imaging, the common complaint was that there's big vessel effects. You're sensitive to all these large vessels. And so just showing this sort of schematized pulse sequence diagram again. Here was our gradient echo. So you collect a 90-degree pulse, and the signal decays exponentially, and you collect your data here, and you get an image, and that's T2 star, what's called T2 star weighted. So it turns out you can do a trick, and this is the Hahn spin echo, as I told you about last week, or last lecture, that if you apply a 180-degree pulse, all the spins that were going at a certain frequency, you can imagine like running around a track, and everyone has a different pace. And suddenly the 180-degree pulse says, okay, turn around and run back. And if they're all running the same pace, they all end up exactly in the same spot at the start line again. So that's essentially what a spin echo is. And so that will refocus at this echo time, which is the spin echo, which is called the spin echo time. And then it'll become defocused again, and then you can apply another 180 and so on, and you keep on collecting. And the signal intensity of these echoes also decays for a different reason, and that's called the T2 decay. Okay, and so this is a classic diagram up above. So this is basically an image just to help illustrate the point of magnetic field distortions. Classic diagram that has been around for 40 years or so, showing the sensitivity of gradient echo versus spin echo, which is R2 star versus R2, to different perturber compartment size, like either red blood cells all the way up to big vessels. And if you're in the range of anything above 15 microns, which is still a pretty small vessel, but still big enough, then you'll have the gradient echo signal will be maximally sensitized to that. Turns out that spin echo, though, will be hardly at all sensitive to that because, and this is the important point that I want to illustrate with this diagram here. I wish maybe I should start working on graphics, but the idea here is that if this perturber is big enough, then spins only really diffuse maybe about 10 microns in an echo time. So if they diffuse 10 microns, then they won't sample that much of a difference of a magnetic field from the beginning to the end of the echo time. So if they don't, then they'll all refocus. It's like runners. It's like they'll all be exactly the same speed coming back to the start line because they're not changing speed. And so then they'll all be refocused. So there'll be no signal. There'll be no dephasing signal that spin echo is sensitive to. So it turns out that between 3 microns and 15 microns, something interesting happens. And that is, to continue the analogy of the runners around the track, that's like if you have 10 runners around the track and you suddenly, 3 runners got really tired and they slowed down a little bit. 3 other runners got an extra boost of energy and they got faster. They won't line up at the start line when they turn around and run back. And the idea there is that with spins that are, if the compartment size is small enough on the order of how much spins diffuse in an echo time, then they'll sample a different magnetic field. And then they'll spin at different frequencies. And then they won't completely realign in that regard. So any changes in that field will also show up with spin echo between rest versus activation. So spin echo becomes optimally sensitive. It's where the spins diffuse on the order of where the field gradient falls off, but no more. And so that's where you optimally sample the magnetic field gradient. So it almost looks like a gradient echo, as if the spins are sitting there. But something else interesting happens when you get really small compartments, and that's the spins are diffusing so fast relative to the size of the compartment. They just kind of fly right by it, and they just average up everything. So it's like as if they're all sampling the same field again. So they all their phase isn't lost. All this is to bring out the point is that gradient echo is more sensitive to vessels of all sizes, and spin echo is sensitive to vessels that are on the order of capillary size to small vessel size. And this is just basically 2 simulations that show the same thing. Like during rest, large vessels, and versus activation, large vessels shows large signal change. Spin echo, it goes back. The signal goes back to an echo, and then there's a tiny signal change. With small vessels, it's still small, but it's on the order of gradient echo. And this illustrates it more practically with real data. I spent time collecting data where every different TR, I shifted the echo time to get super highly sampled curves. And this is a log plot. So these are exponentials, but they look like straight lines. This is simultaneously collected gradient echo and spin echo. And comparing the percent signal change, you see that gradient echo shows a nice linear percent signal increase. Spin echo shows a nice linear percent increase, but it's much lower. And also looking at the contrast, we're looking at the signal difference between these curves. It's illustrated quite nicely that gradient echo has about a factor of 4 higher contrast than spin echo. But spin echo is sensitive to capillaries. This is what I want to bring out here is that the bold signal is most influenced by not only the size of the vessel, that's important, but if you have a voxel that is filled with blood, that's of course going to give you the largest signal because the bold contrast is most sensitive to the blood volume in each voxel. Some capillary voxels might just have 4% blood volume. But if you happen to sample a big vessel, it'll have 100% blood volume. So your signal will be multiplied, 20 times. Your vessel signal will be 20 times larger than a capillary signal, just because of how the voxels sample the blood. I'll talk about a few ways to look around this, but also I just wanted to bring up this paper because I thought it was such a nice paper when it came out. It's still a nice paper. And that is, there's things you can do with the spin echo to grain echo ratio of the signal. So this is basically, I think it was Bob Turner's group at the time, used two things. One, hypercapnia, and two, looking at the spin echo to grain echo ratio. And they found that they were able to make maps of vessel radius in the brain, and also to make maps of blood volume in the brain. And it turns out, as I just mentioned, that the blood volume is a function of, so the blood volume in each voxel will highly weight what your bold signal is. So if you give a global stress, like CO2, that will increase the flow uniformly everywhere in the brain. And the bold signal will only be in proportion to what the blood volume is in each voxel then. So where it's high is where there's a high blood volume. And that's what they found, that they're able to map blood volume, and also within those areas, looking at the spin echo to grain echo ratio, they're able to map the vessel size. And so that was kind of nice. Going back to some older data, this is just basically comparing grain echo with spin echo. First, looking at the baseline image, you see a dark dot there. And that dark dot is because blood has a shorter T2 star than gray matter. So it looks like dark dots, like venous blood does. With grain echo, you have, of course, that's where the biggest signal change is, because that vessel fills a voxel. So that's where the biggest signal change is. However, and here's the interesting part, and this was only at 3 Tesla, with spin echo, you still see a bright area where there's a big vessel. And so it's two things. One, you still have a lot of blood in that voxel, so that's even weighting the spin echo. Even if it's not sensitive to the vessel that's really big, it's sensitive to the red blood cells inside the vessel. So you always have to worry about intravascular signal. Spin echo is not quite as good because of the fact that there's always a little bit of intravascular signal, and that's red blood cells, and spin echo is sensitive to that. So, all right. This was also an early paper that illustrates the same thing. This was by Ravi Menon, in which he was showing basically all these arrows show where the vessels are. And he was showing the echo time dependence, showing that basically where the vessels are, the percent signal change and the contrast is a lot higher. And he was just proposing a way of getting rid of these by just blocking out the areas that show the dark dots. All right, and this also illustrates a point that I was bringing out before, that if you have a voxel and you're sampling a large amount of blood in the voxel, the signal change is going to be very high. So we did a study a long time ago where we basically changed the voxel size from 2.5 millimeters down to 0.6 millimeters. And just using nothing fancy, just partial case-based reconstruction. And what we found is that if we were looking at the histogram of percent signal change versus number of voxels, we find that we have less here, but a lot more of these 25 to 30% signal changes, because you happen to be filling your voxels completely with blood, and that's what fills it up. So that's just a point I want to bring out there. The higher resolution you go, the more you have to really be concerned about these super high percent signal changes, because you're just sampling 100% blood. And this illustrates one more way, just to get it through, and that is, this is a a simulation based on the T2 star of blood as a function of field strength. And it turns out that the T2 star of blood relative to gray matter becomes more and more relative as a function of field strength. This is at 0.5 Tesla up to 4 Tesla. And the intravascular contribution, which is a good thing, at 4 Tesla is about 0.2. It's still high enough to contribute some signal. At 7 Tesla, if you project down, it's going to be about 0.1%. 0.1 or 10% of the signal is intravascular. But so that's a good thing for bowl because there's less intravascular signal. However, it's a great thing if you're doing venography. So venography relies on the fact that if you have a T2 star weighted signal or what's called SWI sequences, susceptibility weighted imaging, they use other tricks along with it, but they rely on the fact that the blood T2 star is less than gray matter. At 1.5 Tesla, blood T2 star is about the same as gray matter. At 3 Tesla, it's a little bit less. At 7 Tesla, it's a lot less. And you can make these beautiful maps of veins just based on the minimum projection of basically the venous blood, because the T2 star is so much less than gray matter. And one of the first SWI images was made by Mark Hickey in 1994. Is that 1994? Yeah. And then there's other methods. It's become incredibly good. There's even more spectacular images here, right here. So, all right, I will continue on a little bit longer, but yeah, I don't want to keep you too long. So with bold contrast, there's many ways of going about modulating the global signal such that you change the blood oxygenation globally to try to find where the large vessels are, find where the blood volume is, so you can actually calibrate it out, or you can get some real information about this. So this was an early experiment where I varied I gave people either CO2 or decreased the amount of oxygen that we're breathing from room air down to 12%. It's not that uncomfortable, but it's just less oxygen. And 21% or 5% CO2 causes this massive vasodilation. So if that occurs, so you can see the signal goes dramatically up with vasodilation with CO2, it goes down with O2, a decrease in O2, because the arteries become now deoxygenated along with the veins. The decrease in oxygen doesn't really affect the change in activation that occurs. It seems to max out the signal such that it dampens the activation-induced signal change. But what's really interesting from this histogram, and this is one thing I found very revealing, is that the change during the stress, and this is everything from 40% signal change with CO2 to minus 40% with decrease in O2, you find that there's a nice relationship between that and the percent signal change with finger movement. And basically all this is saying, like for instance, in certain areas in this image, you had 12% signal change with finger motion. That's pretty high, but it's also where you also had it's also where you had the most change in signal with the stresses. And once again, those are essentially the big vessels where you're capturing all the blood. And you can calibrate your signal by dividing by that big vessel effect to get at more of the capillary effects, if you want to. For most imaging, it doesn't really matter. For layer fMRI, which I'm not going to talk about today, it's a big deal. It does help a little bit. But anytime you calibrate, you're giving up signal. So people like to do other things. So here are the maps from those plots. Essentially, this is activation. This is O2. This is CO2. If you just divide this by this, you get a better map that it's not sensitized to vessels. Okay. I'm trying to find a logical place to stop here. Okay, yeah, I'll stop with just looking at breathing effects. It's going a little bit too long. Okay. So there's other things you can derive from the fMRI signal. And one thing that people always talk about is, oh, well, breathing effects affect the signal. That's an artifact. That's a problem. And it is a problem because typically when you're breathing, the signal changes in some way that might look like bold. It's susceptibility weighted potentially, and it's hard to get rid of because sometimes you breathe in synchrony with the task, or especially if you're looking at resting state, which I haven't talked about yet, you're breathing is at the same frequency as some of the frequencies of resting state. But a few years ago, Rasmus Bern tried to really understand the breathing response function to try to deconvolve that from the time series. It didn't really work that well, but at least it gave us a little bit more information about the breathing effect. So this was a study way back in 2008, where he basically had subjects take a deep breath all of a sudden, like take a huge deep breath and then let it out, and then go along and take another huge deep breath, let it out every 40 seconds, and measured the MR signal that corresponded to it. And it turns out that it was a really robust signal that increased and then showed this massive decrease, this post-undershoot that lasted a long time. And essentially, it filled the brain. It basically is in proportion to the blood volume in the brain. And if he deconvolved this impulse response from this signal, he was able to get the respiratory response function. So that's the kernel that you would convolve with the breathing to predict where the breathing is. So far it makes sense. And it was a beautiful paper with a really nice result. This is various perturbations in breathing, such as breath holding, rate changes where you have them hyperventilate versus not, depth changes, and so on. You can do all these different things. If you convolve this kernel with your time series, you can actually predict the signal very robustly. And they all result in beautiful maps of gray versus white matter. So you can actually derive nice blood volume maps using any of these perturbations. You don't need anything special. You don't need to do a breath hold, which is yet another perturbation. A little bit harder to do. You don't need gadolinium. You don't need breathing CO2 or anything like that. You can just tell them to breathe in certain ways and keep track of it. One thing that I didn't include that I don't want to get into is that the next logical step was to get rid of all the breathing effects by measuring your breath with the bellows and then regressing that out. So, or maybe even trying to do something fancy where you convolve that with the kernel to predict the bold breathing response. We tried everything, and it didn't really reduce the breathing effect. And to this day, we don't know exactly why, but one reason that we suspect is that convolution kernel varies enough over space that you don't really get a good regression. So, but it's interesting. I would love if you had any ideas how to completely get rid of breathing artifacts, because it seems like it should be very straightforward. Measure with the bellows, you get the signal, you regress that signal out from the time series, but it doesn't seem to work. It's interesting. So it's probably a highly nonlinear effect. So anyway, the last thing I want to talk about is the effects of, just for illustration purposes, circling back to the neurovascular coupling thing that I was talking about, that if you actually changed a blood pressure such that there was hardly any flow response, and so I believe Sanjay Kim's group did this, published it in JCBF-FM in 2006, where they, in an animal model, not in humans, they actually added sodium dihydroperoxide, and such that the blood pressure just went almost down to zero, but it was still enough that the blood was trickling through. And turns out that with brain activation, this is with normal blood pressure, the signal responds, pushes through, the bold signal goes out. Turns out during brain activation, when the blood pressure is increased, incredibly low, it goes negative. So that's a direct measure of the amount of oxygen that's being extracted from the blood here. But also importantly, it seems that the neurons still fire nicely. So they're just firing away. They're happy. You give them activation, they fire, they pull oxygen from the blood. It's not so much that they become oxygen starved. And if you bring back the higher blood pressure, the response is just fine. And with blood volume, there's barely any blood volume response. You don't expect it to go negative, but it's just hardly any blood volume response. So I think that since it's already 2:30, I think this seems like a logical place to stop my talk, and then I'll continue on with bowl dynamics. I'll fit this in somewhere before the next talk, and I'll talk about bowl dynamics, and then a little bit of perfusion, a little bit of blood volume, and end there. So the next talk will be probably about 45 minutes long as well. So I tried to cram too much in here, and on top of that, we started late. So I don't know if there's any questions or anything. Yeah.
Just had one question about. I look at the warping effect where basically if you have some kind of, if you choose KY in the anterior posterior direction, see that there's warping image space also in the anterior posterior direction. I was confused as to how does a warping in a direction of K space, which is supposed to be frequency based, translates the same direction in image space.
Yeah. Oh, that's a really good question. It's basically, it's not a direction. It's more a amount of time from sampling with the gradient either high versus then it goes down to 0, then it goes high again. But it's more a function of the time that it's collected. So I have to think this through. I haven't thought about this for a while. Sure.
Are frequencies in space, but there are certain places in physical space, image space, where you're actually misassigning what the frequency that you're reporting in is, and that's what the warping is. So it's not a literal image warping. It's more like what frequency you think you're collecting from in that particular spot. It's not the frequency that you're really reporting at. And so then this ends up, when you do a Fourier transform, looking like an image warping.
So for instance, if you started, so that's a good way of putting it. So over time, you're misassigning different lines of K-space. If you allow more time for the spins to process, different lines of K-space will be misassigned different ways depending on your gradient strength and the order of which you collect those gradients. So essentially, you're allowing different amounts of time for the phase distortion to accrue. So
Study that you were talking about. So the drug that they're using, it has no magnetic effect at all. Or I've always kind of wondered, like, this blood volume is kind of a physical property, but gold is related kind of to a magnetic property.
Yeah.
And I was always curious if the change in gold could potentially be related to
Nitroglyceride it didn't really have that much of an effect. Although I don't know, I have no idea. I mean, that's right, that would cause a significant artifact, but I assume that when they gave it for volume, it might also show up if they're, in a similar way. So, but it was a cool study. Not really and no other groups really have done this. And I think that the normal variation in blood pressure in humans might change the bold signal a tiny amount, but not that much. So it's not nearly about as high as the amount that varies over space from voxel to voxel with big vessels to small vessels when you sample them. So, yeah, okay. All right, well, thank you. Yeah, thanks. And like I said, I will let you know as soon as possible with email when the rest of this talk will be given. And sorry about the setup. I think that we finally worked out what was wrong, and so this won't happen again. Fingers crossed, though.